This is TWiM, This Week in Microbiology, episode 240, recorded on April 15th, 2021. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schechter.
Well, hello there. How are you?
It's April 15th. It's tax day, but we get a break, right? We get a month delay, which is good because I haven't even started mine.
Okay.
Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Did you do your taxes, Michael?
I got them done this weekend.
Wow. And from Ann Arbor, Michigan, Michelle Swanson.
Hello, everyone. Nice to see your faces.
Yeah.
Yes. Did you finish your taxes, Michelle?
Yes, they were finished and submitted, and I filed my quarterly payment.
Nice. Everyone's so efficient except me. Oh, my gosh.
Well, you're busy. You've got public health on the mind.
And I have to do the taxes for the LLC, and it's very complicated for last year. So I've been putting it off. Do you know that when you don't want to do something, you put it off?
Uh-huh.
Or you hire someone. And
yeah.
Sometimes it's the best money you ever spent.
Michelle, I hired an accountant one year, and he screwed it up. So I never hired an accountant again. But I had an English teacher in high school who said to me, Vincent, you work best when you leave things to the last minute. And I never forgot that. Okay, speaking of last minute, Elio, do you know what the goal of a bacterium is?
Yes, I do, but you tell me.
No, I want you to say it. It's your joke, not mine.
It's to become two bacteria.
I love that. So, but we don't say the same for viruses. The goal of a virus is to become thousands of viruses. But they don't have goals, of course. Anyway, we are here on TWiM, and we have a little bit of different episode for you today. But first, we're going to start with a Michael Schmidt snippet.
So today's snippet comes to us from MBio. It was published on the 6th of April, and it's entitled Aspirin Modulation of the Colorectal Cancer-Associated Microbe Fusobacterium Nucleatum, and it's by Brennan, Nakatsu, Galani Camus, Drew, Glickman, Schoen, Chan, and Garrett. And it'll take up more time for me to give you all their affiliations. So I'm just going to jump right in. So as background, we all appreciate from the emerging literature that the clinical correlations and potential mechanistic roles for the microbiome being implicated in the initiation, progression, and survival, especially in today's snippet concerning colorectal cancer, continues and improves with each subsequent publication. We're beginning to understand if there is a role for microbes in cancer. However, with that said, we have not seen a definitive study like we saw for the resolution of C. diff disease, where upon fecal transplantation from a healthy donor, we saw the immediate amelioration of symptoms in patients with the disease manifested by the toxigenic nature of C. diff infections. And I tossed those two papers into the show notes in case you're interested in those. And the reason I bring it up is that the editor of today's paper, Dr. Martin Blazer from Rutgers, published a perspective on the fecal microbiota transplantation for dysbiosis. And really, if we get into the background and significance of this paper, it's really a dysbiosis that is implicated in cancer biogenesis, if you will. And so Dr. Blazer, in his perspective published on fecal microbiota transplantation for dysbiosis, predictable risk, that's in the New England Journal. And I also tossed that into the show notes for you to consider after you read the work of today's authors, because what those three papers do for us is they set the stage. They describe the overarching hypothesis of the work in this field, which is simply put, can we alter the microbiome or the tumor microbiota if you really are going to think small of causing the effect in the patient? And as the authors say, parlay this knowledge into better colorectal cancer outcomes through microbial-informed diagnostic, preventative, or therapeutic approaches. And enter their manuscript, where they demonstrate that aspirin, a drug first introduced to the human race in about 1890, but used in its crude form in folk medicine for many years before. And we know that aspirin is already an established colorectal cancer chemopreventative. And I tossed another resource in, a report from CNN on the history of aspirin and how it can exhibit specific colorectal anti-cancer properties. But today, we're going to look at Fusobacterium nucleatum and the role that aspirin has in influencing the growth of this microbe in culture, in an animal model of intestinal tumorigenesis. And then we're going to take it to people and look in human colonic adenoma tissues. So as the title implies, this is a relatively straightforward paper. Aspirin modulation. So simply put, the aspirin within the system will reduce the concentration of this colorectal cancer-associated microbe in the host. And the hypothesis is thus lessen the risk of developing the cancer. So they walk us through this in a three-part story. They use complementary culture-based experiments where they just simply do growth curves, and it's plus or minus aspirin. They then take us to a murine model of intestinal tumorigenesis, and they finally cap it off where we go to humans with adenomas, and we ask the question whether or not aspirin can work in that particular situation and what they see with respect to Fusobacterium nucleatum. So as background, since many of us are already on prophylactic aspirins because of its anti-inflammatory processes, 2 full-strength aspirins, the big ones, of 325 milligrams each in an average adult, and we all have about 5 liters of fluid for blood, that would give us a blood concentration, if all the aspirin went into the blood, of about 0.72 millimolar or 720 micromolar. And the reason I'm introducing that concentration to our thinking is we're talking about using 10 millimolar concentrations in the growth curves that are the subject of the first experiment. And at the levels of aspirin that they used, 10 millimolar, all the nucleatum strains that they tested, and this was both off-the-shelf American type culture collection. They also tested colorectal cancer strains, as well as this one isolate, FN7-1, which was a clinical isolate of Fusobacterium from a patient with inflammatory bowel disease. And what they showed is that aspirin at 10 millimolar actually dropped the growth curves like a stone. And in fact, FN7-1, the inflammatory bowel one, dropped by 6 log units. You could almost use that as aspirin as a disinfectant to effectively drop Fusobacterium, because the EPA, remember, defines disinfectants that can drop things by 4 logs. They also showed that some inhibitory concentrations of aspirin could also affect gene expression. But since this is a snippet and I know we're short for time, I won't get into that. I'll let you guys take a look at it. So aspirin inhibits growth, but how did aspirin influence Fusobacterium nucleatum in the setting of a colorectal cancer animal model? And so
before you skip over their transcriptome analysis, it was consistent with the idea that the aspirin is acting directly on the bug and changing its gene expression pattern in a fairly major way.
That is indeed true, Michelle. And I was just trying to trim for time. And so in the setting of the CRC animal model, CRC is colorectal cancer. It's a genetically driven mouse model using the APC min-1 model, where FN1 is orally inoculated into conventionally reared animals beginning at 6 weeks of age. So 200 part per million aspirin is fed to the animals or not. And what they were able to convincingly show is that the aspirin completely abrogated Fusobacterium potentiated tumorigenesis. Now, the authors tell us while the data from this experiment were quite compelling, they caution us that the experimental limitations of this model, including the lack of colonic adenomas in either the absence of FN7-1, the Fusobacterium, or in FN7-1 mice on aspirin chow, and the challenges of isolating viable Fusobacterium from the stool of conventional pathogen-free mice left them unable to fully ascertain if their observation was due to aspirin directly altering the Fusobacterium's viability or behavior of the or dominant effects of aspirin on inflammation and colonic tumor development. But they offer that there have been other studies that support their hypothesis that it's really likely the Fusobacterium that's triggering the tumorigenesis rather than the aspirin's ability to confer anti-inflammatory effects to ameliorate the development of the cancer. So far, we've gone 2 parts into our journey. The experiments established the sensitivity of Fusobacterium strains to aspirin. It actually effectively will, if you will, deplete them from their role in causing cancer simply because they're not there in the gut. And the concentrations of aspirin used by these investigators were comparable to those used to study the effect of aspirin on other bacteria and colon cancer cells. And again, a set of experiments I'm not going to describe involve Bacteroides, which is another causative agent thought to be implicated in colorectal cancer, as well as E. coli. But again, aspirin was able to inhibit those. And the final observation or observational experiments take us into humans in order to address the old adage that Alan Dove often likes to say, that mice lie and monkeys exaggerate. So here they take us to humans, and they found that Fusobacterial abundance was reduced in colonic adenomas from patients who used aspirin, even though they were not taking 10 millimolar aspirin. And we are just either looking at whether or not the adenomas that they studied from the 36 human adenoma samples that they analyzed were whether or not they were on the full strength aspirin, which is 325 milligrams, or less than 325, if you will, the baby aspirin that many of us take.
And if I could just interject, this is what we call a retrospective study. So they identified patients and then they queried them and asked, what are your aspirin taking habits? And then parsed them into 2 groups rather than enrolling people and telling them you get aspirin, you don't get aspirin, you get this much aspirin.
Absolutely. And that would be a challenging clinical trial to do because many physicians require their patients to take aspirin as a prophylactic to prevent either heart attacks or to prevent inflammation. And so it would be a rougher time to get that through the Human Subjects Committee.
But we're lucky that they could do a retrospective and get large numbers of people that either were or were not on aspirin. I thought it was very clever.
I agree with you. And the way they did it is they determined the fusobacterial concentrations from human stool samples, specifically looking for Fusobacterium nucleatum, and they normalized it against prostaglandin transporter protein, PGT, which is normally expressed in humans, and the human PGT copies is as a reference for adenoma DNA abundance, and they did that by quantitative PCR. After a data analysis, and as Michelle pointed out, they were then no longer blinded to aspirin use in the patient population. They were blinded when they were doing the experiments, but when they were in the data analysis phase, they were no longer blinded to whether or not the patients were on aspirin or not. And they
and Caitlin, the first author on this paper, is a postdoc who led the work. And she said that was a super exciting day because they had the data sets. They saw that there were clear distinctions between different groups, but they didn't know who was who. So when they got the unblinded information from their colleagues at Pitt, they were really excited to see.
You only need to take one quick look at Figure 6. It slaps you upside the head at the clarity of the data. The aspirin taking individuals are at baseline. They're in the weeds. I mean, it had to be a phenomenal day in the lab. And the other thing is here they observed a 2.3-fold lower mean fusobacterial burden for patients taking aspirin. And then when they compared and contrast if they were on high or low dosed aspirin, they found it didn't matter. It was just the mere presence of aspirin that did the trick. So you don't have to take, a stomach burning a hole in your stomach concentration of aspirin. It's just that because they found no statistical difference between the two. And that was what was remarkable. Again, this was only 36 patients, but again, the load and the precancer adenomas suggest that the aspirin modulation of Fusobacterium nucleatum they describe in culture may actually be occurring in humans. But as they take us to the end of their story, one bacterium does not induce cancer. Their final statement is what's really they need to do next is they have to begin to determine the role of the carriage of the enterotoxic Bacteroides fragilis and or the Colibacterium producing E. coli. And they commented about the challenge of isolating B. frag or E. coli isolates. They have to enrich for them out of the stool in order to find them. And it wasn't as easy to do the quantitation as they were able to do for the Fusobacterium. And finally, they conclude their paper by dropping the gauntlet to graduate students, postdocs, and PIs everywhere by offering that it's critical in how we use our understanding of these microbial host cancer interactions to inform personalized microbiota-based medicine, such as more strongly recommending aspirin to individuals who harbor detectable fecal Fusobacterium nucleatum and therefore may be at a higher risk for colorectal cancer. So we simply could, ask the question, is Fusobacterium in our stool? So that's the snippet.
I'm wondering, as when we sign up for our colonoscopies, those of us in age 50 and older, will we also get give a fecal sample and be told whether or not we have a couple of these bacterial species that have been associated with colorectal cancer?
I think that's a study that needs to be done if it's not being done. And I think we need that diagnostics.
Is it too early for me to start taking baby aspirin each day?
Probably not. If you've had your colonoscopy and you're still clean.
Well, I have a family history, so yeah.
Start the aspirin.
The problem, so both of my parents died of colon cancer, so I have a risk. So I have a lot of colonoscopies, but I took low dose aspirin, and I just was bleeding. I would bump my hands and get clots, So there's a downside to doing that as well. It may not happen in most people, but it is an anticoagulant, right? So I stopped.
Absolutely.
I didn't want to,
Yeah. Wow.
But I think the mechanism, they're beginning to tease at the mechanism, and they look at some of the benzoate resistance, which they think may be an explanation about how aspirin could be controlling the growth of Fusobacterium. So they're beginning to try to tease apart the mechanism, which I think is quite interesting as we begin to look at the microbial ecology of what the microbes are doing in the context of the host. And it's really, fitness. It all goes back to fitness.
I have a colleague at Columbia who works on Fusobacterium. She has interesting results with respect to cancer. She has a mouse model, and she was trained by Stan Malloy, actually.
Oh, wow.
Cool.
We know someone in common.
It's neat that in this paper they have the clinical data, the clinical literature, but then they also have direct impact on pure cultures in the lab. Yeah. So if that's not motivation to figure out how to do genetics on Fusobacterium, I don't know what is. This is a great project. So let me tell you, the first author, as I mentioned, Caitlin Brennan, she's a postdoc in Wendy Garrett's lab. She's from Wisconsin, but she ventured to the East Coast and got her undergrad degree at MIT, where she fell in love with microbiology, thanks in part to Graham Walker. So she did some research in his lab studying mutualism between rhizobium and legumes. But of course, he has such a vast appreciation for microbes that it was contagious. So for graduate school, she went back to Wisconsin because Graham had talked about beautiful work from Ned Ruby and Margaret McFall-Nagai on the squid vibrio mutualism. So she went there and did her PhD with Ned Ruby and Margaret. And then for her postdoc, decided she wanted to take the complexity up yet another level and start looking at the interactions between microbes and the host, but also in the context of a full immune system. So when she arrived at Wendy Garrett's lab, she had never even touched a live mouse. And here, with this set of experiments, it's clear that they're doing that. She was also fortunate to work with some talented colleagues. Daniel Nakatsu came in and did the RNA-seq analysis that we didn't have time to discuss, but showed really clear patterns of changes in gene expression after exposure to aspirin. And then at Pitt, they were grateful for Rocky Schoen's contributions. He's the person that provided the critical human samples that really drove the main lessons home. So she's reflecting on her career thus far, and her advice to junior scientists is to keep trying, but remember to stop and breathe and use your brain. So there's a lot to be said for pausing to think things through when you're facing a problem. So you take time to troubleshoot and maybe think about creative ways, different ways to go from point A to point B. And she says, I can't even count the number of times that she's been doing something completely unrelated, and then this great scientific idea pops into her head and she sends herself an email. So take a break, refresh. It really will help your scholarly thinking.
What is it she said? Take a breath and think?
Yeah.
I like that.
Yeah. Instead of just banging your head against the wall.
Periodically, my watch tells me to breathe.
Yes.
And then it tells me to stand up, but like when I'm podcasting, I can't. So I'm sorry, watch.
Don't let anybody else program that watch. You could find yourself doing all kinds of things.
Really?
Taking out the garbage, making a meal.
Thank you, Michelle. Thank you, Michael.
Thank you.
All right, now it's time to learn about vaccines from Elio. Now tell us the history. Tell us the history before we start doing this, Elio. What's this from?
Okay. Here's the story. I live in a retirement community with 500 other people. This is in La Jolla, California, which is a suburb of San Diego. Lovely place, by the way. Anyhow, we all got vaccinated about a month or so ago. And so everybody became an expert on vaccines. But I found out that most people, being lay people, not being scientists, although educated and sophisticated, had a so-so view of what are vaccines. So I decided to put together a talk called What Are Vaccines? And I gave it 2 days ago. So I thought maybe some of you listeners will be challenged to explain to laymen like your landlady what is a vaccine, and this may just help you. So instead of talking about raw science, a paper, which is what we normally do, we're going to do something totally different today and talk about how do you talk to a general public about a subject like this.
So it's a public service announcement.
Okay.
A public service announcement. So here we go.
All right.
I have a bunch of slides, which
I will show them. And
would be so kind to show. You want to show the first slide?
So yes, and I will just say that if you're listening, we'll put the file on the website, microbe.tv/TWiM, and for others, you could look at it because we'll release this. There you go, Elio.
Alrighty. So could I have the next slide when we're at it? Okay. So this is the cause of death in London monks at the end of the 15th century. So in red are infections, including the first one, which is a mysterious disease nobody knows, but it's probably an infectious disease. The plague, tuberculosis, pleurisy, which probably means pleurisy, empyema. There's a strange disease. Don't ask me what it is. It's probably not an infectious disease. Dysentery, etc., etc. So most people died from infectious diseases. Also, at the time, not only in London, but in every major city in the world, the place was a filthy mess. The streets were full of offal, dead animals, feces, etc. So this was a pestilent place to live in. In contrast with that, the next slide shows you that today, the cause of death by diseases like influenza and pneumonia, which are the major infectious diseases on this list, is way, way down. It's less than, what is it, maybe a couple % of everyone. In fact, if you think about who do you know who died recently, you're going to say not many people died of infectious diseases. So what happened? Next slide. So what happened, the main thing by far was sanitation. The cleaning up the streets was something that happened in the 19th century, roughly speaking, and today we do not tolerate filth on our streets very readily. We might, in some places more than others, but by and large, we expect our cities to be clean. And that caused a huge decrease in the amount of infectious diseases. The next one is vaccination, which is the subject of what we're going to talk about, and antibiotics. So this is what happened. With all these three activities going on, we now live in a world where, though we're buggy, we're totally surrounded by bacteria and viruses and fungi. Most of us do not get much of an infectious disease. We are really quite immune. So anyhow, how do we boost that? By using vaccines. So the next slide, please. Shows you that vaccines are available for a whole lot of things, but not all. So I have a list for vaccines for bacteria, viruses, and no vaccines, alas, for parasites. The bacteria ones include childhood vaccines, which are a mixture. Instead of giving 3 different vaccines, one mixes the 3 into the DPT vaccine, which is diphtheria, pertussis, and tetanus. In the viruses, MMR vaccine, which is measles, mumps, and rubella, are also a childhood vaccine. Smallpox is by itself, but we have a whole lot of vaccines, including many more which are not on this list and which are not usually used, except, like, for instance, yellow fever vaccine, which is used if you're going to travel to a place where there's yellow fever, you might as well get it prophylactically. But generally speaking, these are the main ones. Now, when it comes to parasites, this is where the story breaks down, because malaria, for instance, is a disease that kills about 1 million people a year. It's a big, big deal. And we have no way to prevent malaria. In fact, the Gates Foundation has put in, I think, something like $1 billion in trying to develop a vaccine against malaria. We don't have any vaccines to speak of against parasites. And that is a big, big, big challenge. However, the vaccines that are available, especially the ones which are, why do we give them in childhood? Because we don't want the kids to get sick. Might as well. Why not? Why wait? So anyhow, this is the fact is that vaccines are important and are widely available and widely used. The next slide says, so what is a vaccine? So the next slide says, a vaccine is something that boosts immunity when injected. Now, everybody knows what immunity means, except they don't. Because immunity is, in fact, a complex, not complicated, complex subject matter. Sophisticated, okay. So in the next slide, I think it's the next slide, I explained there are two kinds of immunity. One is called innate immunity, and it's what we're born with, what we're hardwired, and we keep pretty much the rest of our lives. It is not much affected by vaccination, if at all. It's not clear to what extent it's affected. And it consists of the natural defenses, like the surfaces that keep us from having, from the bacteria having ready access to our insides, like the skin, mucous membranes. In addition, we have a particularly interesting defense mechanism in the stomach acid, which kills a lot of the bacteria which we ingest. And there are blood proteins, which you all know what complement is. Complement is a mess of a collection of proteins, which I did not try to explain to a general public. I would not try to explain it. I would simply acknowledge that some people may have heard of complement. Anyhow, these are, so besides the integuments, the surfaces, and the proteins in the blood and in tissues, we have white blood cells. White blood cells are leukocytes. That's what it means. Leucum means white and cyte means cell. And they're found in the blood. They're found in lymph nodes and in vessels. And they cause inflammation, that is pus. So inflammation is a big deal. It happens to all of us. All we need to do is to scratch our skin and it gets inflamed. Anyhow, these are natural defenses which all of us are born with. If we're not born with, we suffer from a very severe condition of one sort or another, an immune deficiency, innate immune deficiency, which are fortunately very rare. So these are wonderful mechanisms to keep us alive in a buggy world. However, we can change this by using vaccines. And the next slide shows you what is called acquired or adaptive immunity, which is something which is different in everybody. Everybody has a different set of antibodies, antibodies being the proteins in the blood and in the tissues, which are made as a response to an antigen. I try to explain what that means. What is an antigen? An antigen is a foreign substance, a protein, a polysaccharide, whatever. And this causes arises the production of specific antibodies. There are about 5 million possible combinations, so we're not going to run out of combinations, but everybody has a different repertoire of antibodies in their bloodstream.
Depending on what they've experienced so far in their life, right?
I'm sorry?
depending on what they've experienced, what they've confronted.
Depending entirely on their experience.
Yeah.
And it can be spontaneous, that is, we're in contact with germs and we acquire disease. Now, you may have heard of the hygiene hypothesis, which says that kids who are too separated from bugs in childhood are in trouble because they do not develop this kind of adaptive immunity. And that may be true. So anyhow, there are two kinds of adaptive immunities: spontaneous, which is simply we live in a buggy world, we're kind of in contact with it. The moment we go through our birth canal, we acquire a whole bunch of bugs, or artificially done by vaccination. And this changes with time. So everybody here is different, whereas the innate immunity were kind of similar. Okay, so this is the two kinds of immunities that you have to know about, and vaccination is involved in boosting or in establishing acquired adaptive immunity. So what are antibodies? The next slide shows that antibodies, which you know, but my buddies in the place where I live may not know, are also known as immunoglobulins or gamma globulins, and they're produced in response to a specific antigen. An antigen combines with the antibody and recognizes, the antibodies recognizes the substances being alien, such as bacteria, viruses, foreign substances. Now, the drawing shows you what a diagram of the coronavirus or COVID-19, and it shows it's got spokes. Those spokes, which everybody has heard about, is what the vaccines are trying to hit. And in blue, what you see there is a schematic of antibodies. Antibodies are large proteins made up of subunits. They are large subunits, which is the large blue bent drawing, and small subunits, which are the tiny ones. So what they do is at one end, at both ends actually, in this Y-shaped molecule, they attach to the antigen. In this case, if you have antibodies against the spoke proteins, they will attach to the spoke and neutralize it. That is, if you have antibodies stuck to the spoke, you're not going to have an infection because the virus cannot attach to what is known as
the image of a lock and key is useful here. So the antibody is like a key, and if it finds its particular lock, it will fit and work against it. It's very specific.
Okay. So this is what an antibody is, and vaccines make antibodies. Okay, so the next slide says a little bit about the history of vaccination. It turns out that in Africa and Asia, for many, many centuries, people were inoculated with bits of smallpox. Smallpox was a big disease. In fact, it killed about half the people who got it, something like that. But if you took a little bit of smallpox material from a sick person and you put it under the skin of a healthy person, a few died, about 1% died, but the rest got a mild case of smallpox, and it turns out they were immune to further smallpox. So even though a few died, without it, half died. So 1% is a small price to pay considering the risk, which is 50%. So that was what was going on in Africa and Asia. The procedure was called variolation because variola was the name for smallpox. However, Jenner, who was a physician in England, in London, in the 1700s, realized that milkmaids, that is, women who milk cows, did not suffer from smallpox. Instead, they got a disease called cowpox, which is a mild disease, which is a disease of cows. It does not kill cows particularly, and it certainly doesn't kill people. So if you take, the word vaccine comes from vacca, which is Latin for cow. So vaccination is to make somebody a little bit into a cow, if that's what you wish.
And cowpox and smallpox were caused by two different similar viruses, correct?
They're two different viruses completely. They are, they're related, but they're completely different in that the smallpox virus kills people, the cowpox virus does not. So Jenner said, let me use cowpox material as the source of a vaccine. Call that a vaccine. And he did. So a lot of people, including us, get cowpox virus when we're very young. A form of cowpox virus. By now it's been modified a number of times, but in the early days, it was pure cowpox virus. We got infected, and in fact, a lot of people got it on the forearm, on the deltoid, on the side of the arm, and got little lesions, and many people have scars, tiny scars, which are visible usually on the right side. That is the history of vaccination in its origin. It started that way it was widely used from 1800 on up. In fact, it became legally binding to have kids vaccinated. You had to do that.
And in fact, I saw a cartoon on Facebook this week with a little kid pointing at his mother's variole or cowpox scar. And the child asked his mother, "What's that?" And she said, "It's a vaccination against smallpox." He says, "Why don't I have one?" And the mother's response was, "It worked."
Great.
Herd immunity.
Nice.
Herd immunity.
Nice.
Anyhow, with the beginning of microbiology, roughly 1850 or thereabouts, bacteria got to be understood and were seen to be cause of diseases. People realized that they could make vaccines. And one of the very first one was actually by Pasteur. He was working with the anthrax bacillus, and he decided, gee, I cannot inject anthrax bacilli into people, but maybe I can make a debilitated strain, a lesser strain, which means I can inject it, the equivalent of cowpox versus smallpox. So he grew anthrax bacilli at high temperature, which for reasons I don't still know to this day why that is, but it becomes debilitated, and injected that into a 4-year-old kid, and the 4-year-old kid was then exposed to anthrax and didn't get anthrax. So he was immune. He was vaccinated against it. And at that time, people realized that this was true for other bugs. And as the development of microbiology happened in the second half of the 19th century, more and more vaccines were made. And so a whole collection of vaccines became possible. The next slide shows what kind of vaccines do we have. So we have unrelated but not dangerous germs like cowpox versus smallpox. We have live but attenuated like the anthrax bacillus. By the way, it's really remarkable. Pasteur was a chemist. How he got into this, I don't know, but it's part of his genius. Anyhow, another attenuated vaccine is the polio vaccine by Sabin, as opposed to the Salk vaccine, which is inactivated agents. This polio vaccine by Sabin is live viruses. The polio vaccine by Salk is dead viruses. And the MMR virus, that is measles, mumps, and rubella, are also debilitated viruses, attenuated viruses. Now, let's spend just a moment comparing and contrasting them. What's the point? Well, if you have an inactivated agent like polio, it's going to be safe because it's dead. Obviously, you can make a mistake, which happened in its history, and you can, in fact, give people live virus, but that's a mistake. It's not supposed to happen. And so if you give the Salk vaccine, you're not going to give anybody polio. Whereas if you give the Sabin vaccine, since it's a live virus, it may be that it could become reactivated, and this has happened. On the other hand, the advantage of a live but attenuated vaccine is that if you give it to a few people, it would spread across the population. So you don't have to vaccinate everybody, just have to vaccinate a certain number of people, and it will spread around the population, and everybody essentially will become immune. That's so there are advantages and disadvantages. A lot of argument went on between Salk, Sabin, and their partisans, and it's probably still going on, I think. Anyhow, then there's a thing called toxoids. So what is that? Many bacteria, tetanus for instance, Clostridia, many other, make toxins That is, they make poisons, compounds which they secrete into the body and which can kill you. In the case of tetanus, the tetanus toxin is extremely dangerous. However, if you look at the drawing, on the left side of what looks like an ear, a red ear, that is the portion of the toxin which is toxic, which is the toxin moiety, the thing which makes you sick. However, if you remove that somehow chemically, if you modify it, which is easy enough to do, you don't change the rest. The green part, the other ear, is the thing which is antigenic. That is, that makes an antibody against it. So what you're doing is you're reducing a toxin to a toxoid. A toxoid will be antigenic, therefore will elicit the formation of antibodies, but will not be dangerous. And there are toxoids against a number of toxins. So this is the way that the kind of vaccines we have. Now we get tetanus toxoid injected several times in our lifetime because, as in the next slide, how long does it last? Well, usually it lasts for years. However, it doesn't last forever. So if we have a infection, let's say we fall on the street and we cut ourselves and we go to the doctor, if we're adult enough, we're going to get the tetanus toxoid shot. It's a booster shot. That is, we've already seen, our body has seen the toxoid before, has made antibodies to it, but the antibodies don't last forever, so it makes more. So usually the immunity from vaccines lasts for years, but obviously if something is just a year old, like COVID-19, we don't really know how long it lasts. There's several notions that it will last a while, but we don't really know until the time passes, right? So how come vaccines last a while? It's because there are cells in the body, and I did not explain this in any great detail. They're called memory cells, and they continue to make antibodies for a while. So as long as there are memory cells there, we continue to make antibodies. So vaccination lasts a while because the memory cells keep working. However, booster shots goose up the memory cells to make more antibody.
And
so
Elio, if you remember on that one TWiM that we did, arguing for why it's so important to get your measles vaccine, is if you get measles, the measles virus kills off memory cells.
That's right.
And that's why it's so important to get your measles vaccine, because you can literally wipe out all of your memory, and then you need to get reacquainted with all of the old vaccines.
And this is why measles, which we consider to be a childhood disease, like smallpox, was one of the reasons why the Native Americans, when exposed to the Spaniards and their viruses, died by the droves. In fact, they were decimated. Decimated means that 90% were killed. And there were two viruses which are involved. Smallpox, which we understand. The Europeans were more or less immune to smallpox. But measles sounds funny because, as I say, it's usually considered to be a childhood disease. But like Michael said, it is not a mild disease at all. It is a very serious disease, and getting immunized against it is a very important thing. Thank you very much. Okay, the next slide talks about the different choice we have. We have a choice called passive immunization. Instead of getting an antigen, which is a way of telling the body, make an antibody against it, we can get the antibody. So as in fact, most of my fellow residents here have heard, antibodies are present in convalescent serum, that is serum from people who had gotten sick and got well. So they were exposed to the antigen, they make antibodies, and the antibodies are going to be around for a while. This is what was given to Trump when he got sick. He was given a gamish of antibodies from convalescent serum. That's one thing you can do. It's a little bit tricky because you have to clean up the antibodies. Otherwise, you're going to give people a lot of strange antigens, and they're going to get things called serum sickness. Anyhow, but you can also make antibodies in the lab, and monoclonal antibodies, which, by the way, in the question period, somebody asked me to explain monoclonal antibodies, and frankly, that ain't easy to do to a lay public, but I tried. Anyhow, you can make antibodies in the lab, and therefore you can purify them, and you don't have the problem of having to inject a whole gamish from convalescent serum. So this is a choice that one has. The next slide shows this. Now I went into what are the vaccines against COVID-19. So there's a whole collection of them, and everybody has heard of this. By the time I gave my talk, everybody had been vaccinated, and everybody's an expert on vaccines.
Good for them.
So they all got the mRNA vaccine in oil bubbles, Pfizer and Moderna vaccine. That is one of the most incredible histories in the history of medicine, that the development of a vaccine happened that fast. Normally, it takes years and years and years. This was done in a question of months. And so mRNA vaccines are something that, frankly, I had not heard about a year ago. But I'm sure Michael had, but and probably Michelle did. These are a strange concept that was lying around, and some people at Pfizer and Moderna resurrected it. Now, mRNA is very sensitive to ribonucleases, to enzymes which destroy it. So if you just injected mRNA, it wouldn't last very long. So you put it in oil bubbles, and within oil bubbles, it's going to last a while.
And that's what took the time. It's the development of those lipid nanoparticles. And the lipid nanoparticles was about 15 years of very, very rigorous research by many groups, funded by the NSF, funded by the NIH. And funded by companies. And that is really how they were so quickly able to put, if you will, a recipe card into an oil protective bubble that they gave to our muscle cells.
Now, the fact that that's handy, because today, if you want to make a different vaccine, like you had in mind to make one against variants, you could make it very easily. It's a quick thing to do. You can also put DNA from the complementary DNA to the RNA of the virus in different virus carriers. And AstraZeneca uses chimp adenovirus, and Johnson & Johnson use human adenovirus. I explained a little bit what that means. You can also use a killed whole virus, and there are 2 Chinese vaccines about which we know nothing, which are done that way, or use just a virus subunit, in this case a spoke protein. And there apparently Novavax does that, and there are 2 Russian vaccines like that. You could also, in theory, attenuate a live virus. We don't have an attenuated live virus for COVID-19, so we don't have that yet. But there's a whole variety. So there's a whole collection of different vaccines against COVID-19. The next slide.
Let me just make a correction. I'm sorry. The Chinese vaccine, one is inactivated, one is adenovirus vectored. And the Russian vaccines are both adenovirus vectored.
Oh, I'm sorry. I missed that.
No, it's okay. Not so important.
They're not going to kill me.
No.
We won't let them.
Okay. Now, a thing that always comes up is how come influenza virus, we need to get a new shot every year. And I try to explain that. It's unusual. And it's not likely that we need a different vaccine for anything else. Hardly anything else means that we need to get a different vaccine. How come influenza virus? So the influenza virus is kind of unique. It's called a segmented virus, meaning that its genome is not a single molecule wrapped around inside a protein shell, which is what everybody has seen. It's actually made of different segments. That's why it's called segmented. And there are 8 or so segments in influenza virus, and they're shown on the top in green. So the different sizes, they're lined up there. Obviously, inside the virus, they're not lined up at all, but they're drawn that way. Now, what happens is that the influenza virus is not just a human disease, but it's a disease of pigs, horses, dogs, a whole collection of animals. So when a new year comes up, that is, we start a new year, and we start in this southern hemisphere, which is when it's warm, when it's cold here. So in the southern hemisphere, we find the virus that's going to come north. And that virus is usually made by a mixture of different viruses. So if you're on the left, if you go down the line, you see the genomes in different colors, in green, red, and purple. Each one of those viruses is different, but they can get together and make a new virus, which is a different gemisch. So at the right, you have a virus which has some of the genes are made up of purple, red, green, etc.
And that can happen if a farmer has chickens, for example, and pigs that each have the virus. If the farmer gets infected with different kinds of flu viruses, they can commingle within the farmer's body and then create these new viruses, human viruses.
So the upshot is that every virus, every flu virus, every year is going to be different. And therefore, you need a new shot every year. You may need to make a new vaccine. By the way, there's a race every year. People who are in the vaccine business go to the southern hemisphere, try to pick up as early as they can what is likely to be the popular virus in the north and make a vaccine out of it as fast as they can. Doesn't always work. This is why the flu vaccine is considered to be very effective some years and less effective other years.
And most of the flu virus vaccines are grown in chicken eggs. And so
just a minute.
All right, I'll shut up. I haven't seen the slide, so I don't know what's coming.
Okay. So
I don't know what's coming.
The next slide is the news. First of all, there's a new vaccine which has a different molecular design. It happens that the spoke protein is a very flexible protein, and it changes a lot. So a new vaccine is made on the basis of a new kind of spoke protein, which is less changeable. And this is made by adding proline, 8 proline molecules into the protein that makes the vaccine. So we have a better vaccine in the making. But like Michael appropriately said, COVID-19 can be grown in fertile chicken eggs and can be used for a vaccine. Now, this is where the flu vaccines are made. They're made in chicken eggs, which are fertile. Now, fertile chicken eggs are something you can buy in the store. And everybody in the business, in the world, in the business of making vaccines can get chicken eggs, fertile chicken eggs, inject them with the virus will grow, you harvest this, you get a lot of virus. And having a lot of virus means you have a lot of vaccines. And it turns out that the COVID-19 virus gives a yield of 5 to 10 times greater than the flu virus. It's just wonderful to grow it in chicken eggs. So this is everybody can make can do this. Injecting flu viruses in chicken eggs is that easy. Everybody can do it, and this means that many countries can make new vaccines. So this is news. So that's what I told them. And next slide.
Can I just make a correction? I'm sorry. So this vaccine is Newcastle disease virus. And that's the virus they put the spike in, and that grows in the eggs. So SARS-CoV-2 does not grow in eggs, but this
I'm sorry.
Newcastle,
I messed up.
Newcastle grows in the eggs because it's a chicken virus, and that's the carrier. And you're right, it grows really well. This is being done at my former department at Mount Sinai in New York City. Okay.
So that means it's safer too then. You don't have to worry about having large quantities of it.
No, but you might grow feathers.
Virus.
You might grow feathers, Michelle.
Well,
so as of last Monday, there's evidence that short-term vaccine with a single dose works. Now, a dose of Pfizer or Moderna. Now, this is handy because this doubles the amount of vaccines that exists.
Yeah.
Instead of having 2 shots, you take only 1 shot. And so it looks like a single dose of Pfizer, Moderna, and probably any of the other vaccines is enough. Certainly the Johnson & Johnson vaccine, 1 shot seems to be enough. So this is the news that I told them. I thought this was one way to reach a broad public. The reaction I got was very favorable. People said, yes, they understood it. I didn't test it. But I think that people apparently enjoyed it. And so this is one way in which you can, if you like to use this, it'll be in these slides will be in the show notes, and you're free to use them any way you want in explaining to your landlady what is a vaccine. So thank you very much.
Or
Next
landlord.
slide.
Or landlord.
Landlord. Right. Next slide. Thank you. I thanked everybody. So that's it. You can get rid of the slides if you want.
Hey, Elio, what was the best question you had?
What's that?
What's the best question they asked you?
Oh, gee, I don't remember. I didn't get that many questions, and they were not great. I must say, they were fairly simple.
Okay. Very good.
What was the best question? Oh, yeah.
That's all right. No worries. I think you probably scared them because you're so smart.
You covered a lot of history, though. That's interesting.
That's good. All right. Let's wrap it up. That's TWiM 240. If you want to find the copy of this slide presentation, the show notes are at microbe.tv/TWiM. If you have any questions and comments, send them to twim@microbe.tv. And if you like what we do, consider supporting us, microbe.tv/contribute. Elio Schechter's at Small Things Considered. Thank you, Elio.
My pleasure. Thank you.
Michelle Swanson's at the University of Michigan in Ann Arbor. Thank you, Michelle.
My pleasure. Everybody stay safe. Hang on for another month or 2.
Michael Schmitz at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Michael, do you take aspirin?
I do.
Very good.
I've taken big aspirins because my healthcare team believes in anti-inflammatories.
Oh, okay.
So I've been taking a big aspirin.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.